How can you get the most accurate functional characterization of your omics data? Enrichment analysis is the standard approach, but with so many methods available, it's hard to know which one is best suited for your dataset. That's where our generalized benchmark for all types of enrichment analysis methods comes in. I'm a PhD student at Stockholm University and this work was supervised by Eric Sunhammer. Functional enrichment analysis (EA) is a widely used approach to interpret genome-wide functional genomics experiments. EA condenses differentially expressed genes into overrepresented biological functions or processes, providing a system-level understanding of cells and potential disease biomarkers or therapeutic targets. EA utilizes gene expression profiles from DNA microarrays or RNA-seq, typically obtained from case/control or treated/untreated experiments. Functional gene sets are defined by resources like Gene Ontology, Kyoto Encyclopedia of Genes and Genomes, Reactome, Dysgenet. There are plenty of EA methods, yet it's unclear which one works best. The reason for this is that previous benchmarks were not designed for all categories of methods, used a small subset of known diseases, mainly cancer-related, used scoring metrics that are unadapted to benchmark sensitivity or biased towards some categories of methods. EA methods can be grouped in 4 categories: overlap-based analysis methods, OVA, that test the proportion of differentially expressed genes and gene sets, per gene scoring analysis methods, PGA, that assign scores using sorted fold changes of all genes, pathway topology analysis methods, PTA, that use pathway databases to weigh gene importance, and finally network-based enrichment analysis, NEA, methods, that evaluate differential expression in a functional association network. All 4 approaches were run on experimental gene expression datasets and on 2,460 random datasets. We collected 82 case/control studies from Gene Expression Omnibuses curated in JAMA for 26 diseases, of which only 13 are cancer-related. The Kyoto Encyclopedia of Genes and Genomes pathway database was used to evaluate the EA methods. In the previous benchmarks, the target pathway was treated as the single true positive and all other pathways were assumed to be false positives. We introduced the disease pathway network to counteract the shortcoming of the single target pathway approach and improve the sensitivity assessment in an unbiased way. We connected 92 disease pathways into a disease pathway network based on gene overlap and network connectivity, using the 20% top scoring links in HumanNet. We found that these pathways are highly interconnected, with 88 significant relationships per pathway, on average. As this amounts to around 30% of all pathways in the database, we also assess the Go-semantic similarity between each target pathway and the significantly related pathways to select the closest ones overall. Here is an overview of the disease pathway network, highlighting the diseases and subclasses included in the benchmark. The network illustrates the coherence of our disease pathway model, with stronger connections, represented by larger edges in the figure, observed between diseases within the same subclass. The figure shows the absence of a cancer-focused dataset, overcoming another previously identified limitation in benchmarking enrichment analysis methods. In this study, positive and negative benchmarks are independent. To address the imbalance of positive and negative pathways, only target and target-related pathways from the positive benchmark were used to calculate true negatives and false positives in the negative benchmark. Then, we extracted TPR, or sensitivity, and TNR, or specificity, for 14 methods. To condense the method performances in one balanced, comprehensive and robust summary index, we use the geometric mean of true positive rate and false positive rate. To emphasize the significance of capturing true positives in the top predictions, we calculated the median relative rank of true positives after sorting by p-value. Network enrichment analysis methods outperformed all other EA approaches. NEA methods differ from other approaches by considering the connectivity of differentially expressed genes with functional gene sets in a network context, rather than assuming gene independence. The better performance of NEA methods may be attributed to their ability to address the limitation of low database coverage through the utilization of genome-wide functional association networks such as FunQ and String. In this benchmark, OVA remains a very conservative approach. PGA and PTA perform overall well in only one of the 2 scoring metrics. We also assess the performance of EA methods on randomized data. An ideal method would produce a uniform distribution of p-values ranging from 0 to 1 for each pathway when randomized data is used, with 5% of the p-values falling below the 0.05 cutoff. We observed that OVA methods in camera were highly conservative, as all of their p-values were below the cutoff. ROAST shows the highest median value. GSVA, Binox and Neat with string have intermediate median values, while the rest of the methods have lower and quite reasonable values. The heat map shows the false positive rate of the average top 10 skewed pathways predicted by network-based EA methods. The top 10 skewed pathways towards zero share a common characteristic: they have a high fraction of intralinks and large fraction of high-degree nodes within the functional association network, indicating that these pathways are isolated and exhibit community-like properties within the network. In these circumstances, methods that apply statistical properties of crosstalk generically to all pathways like Binox and Neat, lack the ability to adapt to the distinct characteristics of individual pathways. In contrast, Anubix accurately predicts the expected degree of crosstalk for each specific pathway by preserving the unique properties of each pathway, and thus minimizing the bias towards those pathways. With this, thanks for your attention. Please, stop by my poster and let me help you choose the best method for your data. Otherwise, send me an email if you are interested in having more details about our EA benchmark.